Visual Place Learning in Drosophila melanogaster by Tyler A. Ofstad, Charles S. Zuker, and Michael B. Reiser. Both humans and animals use spatial and visual cues to direct their movements. For example, honeybees and ants are able to return to their hives and colonies after traveling great distances to find food. But how do other insects, for example, fruit flies, use these cues when they have no specified home location to return to? Tyler Ofstad and colleagues sought to answer this question in their research. To test the fruit fly's ability to form spatial memories, an intricate spatial field consisting of multiple tiles laid out in a mosaic pattern was used. The design was inspired by the Morris water maze, as well as a heat maze designed for cockroaches and crickets. To change the temperature of the tiles, the investigators heated up all of the tiles to 36 degrees Celsius, which the flies dislike, and cooled one of the tiles to 25 degrees Celsius, which the flies enjoy. In addition to these spatial cues given by the tiles, the scientists surrounded all of the tiles with LED lights, which could generate different orientation patterns. The visual panorama consisted of vertical, horizontal, and spiral patterns. The flies were conditioned to this environment by fixing the location of the cool tile in relation to the visual orientation patterns. The scientists then tested the fly's spatial memory formation by rotating the visual cues 90 degrees clockwise or counterclockwise, as well as heating all of the tiles to 36 degrees Celsius. As a result, the mosaic platform no longer had a cool tile, and all of the visual orientation clues were different. The flies searched for about 1 minute for the cool tile in its former location. These results demonstrate that the flies were able to use spatial temperature and visual orientation clues of the LEDs to form a memory of the location of their preferred tile. To ensure the flies were not influencing or following other flies to the safe tile, single flies were trained and tested individually, showing equivalent results. Further testing concludes that the flies only retained these memories for up to 2 hours. The investigators were also able to isolate the part of the fly brain that is responsible for this type of memory formation. They found that the ellipsoid body, which controls voluntary movement, also seemed to control spatial memory formation. By using fly lines that contained silenced neurons in the ellipsoid body, they were able to show that visual place learning was severely impaired. In conclusion, Drosophila can learn and recall spatial locations in a complex environment with remarkable efficiency. No other updates of this article have been found.